All right, sorry about that. So we'll try and pick up where we left off from yesterday. From last class. Last class, we spent time talking about the basement membrane, talking about what proteins consist of the basement membrane. We spent some time defining glycoproteins versus proteoglycans, fibrous proteins, and GAGs. A lot of this two-part lecture is going to be things broken up into twos, basically. And so we first talked about the basement membrane, also referred to as basal lamina. We're now going to pivot and talk about the connective tissue ECM. So in yellow on the top right here, I'm going to use my mouse and not try and wander over. We have in yellow the basal lamina, or the basement membrane. That's usually, again, situated underneath a sheet of cells like the epithelium or like the endothelium or the inner lining of a blood vessel. What ends up happening here is that with the connective tissue ECM, it's going to be situated beneath, at least in the case of skin here, beneath the basement membrane. And a lot of times it's going to also surround the basement membrane as well in some instances. The connective tissue ECM is going to be comprised of many protein fibers that are embedded in this mixture of proteoglycans. So you can think of it almost as this gel-like structure with fibrous proteins woven with all throughout it. A key distinction between the basal lamina and the connective tissue ECM is that, again, you have a little bit of, for the basement membrane, you have a small amount of protein structure that sits underneath many cells. For the connective tissue ECM, you have a large amount of protein content with fewer cells interspersed within it. And so what's a key thing to note here is that with connective tissue ECM, it's going to help determine and define what the physical or mechanical properties are of a tissue. So when it comes to the production of the connective tissue ECM, there is one cell population that's typically both within the connective tissue ECM and responsible for producing it. These are called fibroblasts. One thing that's unique about fibroblasts is that you have them in basically every tissue in your body. Again, because you have the ECM in every tissue in your body, there's going to be unique fibroblasts that are in every tissue in your body. So they are the cell type that is the predominant cell type in connective tissue and the predominant cell type that is producing the ECM. They're producing fibrous proteins that provide strength and organization to the tissue. They're also the cell population that expresses and produces GAGs and proteoglycans. A key thing to note that we're going to spend some time talking about is some of the properties of GAGs and proteoglycans and how their properties contribute to some tissue, how their qualities contribute to tissue properties. So again, GAGs, glycosaminoglycans, they are these disaccharide two-sugar repeating units. They're typically going to be like there's going to be, the first sugar is going to be some sort of amino sugar followed by uronic acid sugar. And you can see in hyaluronan what those repeating sugar units look like. We have N-acetylglucosamine and then glucuronic acid. What's going to separate, the key thing to note here is that what's going to separate the species between different kinds of GAGs are the kinds of sugars that are within them. There's going to be different kinds of linkages between those disaccharide units. And when we say different linkages, what I mean there is that there's going to be bonds between different carbon groups between the adjacent monosaccharides here. And another thing that's going to distinguish each GAG is going to be the number and the location of sulfate groups. The reason why I say sulfate groups is because with the different kinds of GAGs, the four main groups that we have here, one is going to be hyaluronan, but then we're going to have chondroitin sulfate and dermatan sulfate, heparin sulfate, and keratin sulfate. So you can tell by their names that a fair number of them have sulfate groups on them. And again, what's going to distinguish them is how many sulfate groups are there and where those sulfate groups are located on the molecules. The key thing to walk away and take away from this slide is that we have qualities or features that define GAGs. They have repeating disaccharide units. They're all going to be an amino sugar and a uronic acid. And what distinguishes them, what is specific to their species of a GAG is what sugars there are, how these sugars are combined to each other, and how these sugars are modified especially by sulfate groups. So what's important to know is their conformation is really important. They typically like to occupy and have long extended conformations. They like to occupy a large amount of volume relative to their mass. One way I like to think about this is the annoying person on the UTS bus who occupies a lot more space than they probably should is kind of how GAGs behave in tissue. They occupy way more space that relative to their mass. I know you all are thinking about that one person that you typically see on the bus doing that. So I thought it was a lighthearted way to talk about it. Clearly it didn't work, so we'll keep going. So with GAGs, another thing to note is that they have an incredibly important negative charge that's associated with them. That negative charge is really, really important because what ends up happening is that the negative charge associated with them helps contribute to their function. So in addition to their negative charge, that's one important feature. Another important feature that helps contribute to their function, I should say, is that there's a high osmotic pressure that helps facilitate the GAG's function. So what I mean by that is usually we find high amounts of GAGs in our cartilage. So for example, when you are undergoing compression in the cartilage in your knee, what ends up happening is this GAG that has a high volume to mass ratio, when it's compressed, that negatively charged molecule, all of their negative charges are going to be all of a sudden in close proximity to one another. And so there's going to be a repulsion. You're going to have negative charges adjacent to negative charges, which means those negative charges are going to want to repel each other away. So that's going to help with resisting compressive forces in tissue like cartilage. And in addition to that, that's one force that helps withstand compression. Another force that helps withstand compression is osmotic pressure. Once we have, what I should say is that there's a lot of, that there is a fair amount of water content in tissue that has a high GAG content. So in your cartilage, when there's compression, there's going to be the pushing out of water that then there's going to be an osmotic pressure to drive that water back in to resist that compression. So the two key features here that facilitate GAG's function in withstanding compression or responding to compression is that high negative charge, as well as this high osmotic pressure that drives water back in to resist compression, particularly in cartilage. This is just this is just to help provide a little bit more information on GAGs, not asking you to memorize all of this, but just to help give you a little bit more content, more information about each of them. We have chondroitin sulfate, which is the most prevalent GAG. We have heparin, which has a high negative charge density that's present in a lot of cartilage tissue, but also is what we call a growth factor depot. That's one thing that's particularly unique about heparin. When I say growth factor depot, what I mean by that is whenever you see a heparin binding site on some protein, heparin is able to bind to growth factors, these signaling proteins that can really alter cellular behavior. And so what that means is if we have some fibrous protein and then heparin comes in and binds to that fibrous protein, on heparin, we can have growth factors that come in and bind all over heparin. And now growth factors that are usually just released into soluble bodily fluids like blood, like the lymphatics, can be anchored down in the tissue and easily accessible to cells like fibroblasts that are in tissue that don't always have immediate access to what's in the blood, but can now access growth factors that are anchored down in the ECM. And so, and again, hyaluronin was the main species of GAGs that we saw without sulfate in its name because it's the only non-sulfated GAG here. So proteoglycans, again, an important distinction between proteoglycans and glycoproteins. Easy to confuse the two because it's basically taking two parts and kind of swapping where those words are associated. The suffix is what comprises most of that molecule. Glycoproteins are mostly proteins. Proteoglycans are mostly sugars. So, and when I say mostly sugars, proteoglycans can contain 95% of their molecular mass can consist of carbohydrates or consist of sugars. And so that usually means that there's a small amount of protein with lots of GAGs or lots of sugars added to that core protein. And so what we have, the way those proteoglycans are produced is that there's first some core protein that is expressed and translated in the cell. And what ends up happening is that within the ER, or within the Golgi, forgive me, that's the organelle where glycosylation happens. Glycosylation is the process of adding sugars to a protein. And so as an example here in this image, we have a core protein where at a serine amino acid or a serine residue here, we have this GAG that's added, many sugars added to this amino acid residue on this core protein. And so it's going to happen at specific sites, and it's typically a really what's an interesting feature of this process, not important for you guys, not something that you won't be assessed about, but these glycosylation events, these addition of sugars can be really unique from tissue to tissue and can go wrong in disease, and is now like a huge area of research to understand how does glycosylation of certain proteins alter disease, or how is it important in just normal biology. There's a lot to understand here. And so this is the key thing to note. The key thing to walk away here is that glycoproteins have mostly protein with a little bit of sugar on it. Proteoglycans have way more sugar modifications, and those sugar modifications can vary based in disease or normal biology. One example is decarin, which has a core protein that's situated here to the left, with only one GAG chain on it. So, you know, this is an example of like a very small amount of sugar modifications to it. Whereas aggrecan is a core protein with over 100 GAG chains on it. And so the amount of glycosylation, or the amount of sugar molecules, can vary, but then it's going to be relative based on the size of that core protein. And so you can see with aggrecan, that core protein is a lot larger, which means there's a lot more opportunities for sugars to be added to it. And so the takeaway here is that there's lots of variation in the size of these proteoglycans based on typically the size of that core protein. A key thing to note is that the proteoglycans that are synthesized by fibroblasts serve a really important function. They can contribute to the assembly of the ECM and how it's organized. There's also the ability to regulate and traffic molecules. We talked about some GAGs having the ability, chiefly heparin, to serve as a growth factor depot. So it can regulate and trap certain growth factors or signaling molecules. It can also change the size and charge of a porous network. Again, bind chemotactic signals, meaning it can bind and anchor at growth factor binding sites, certain signaling factors here. And a key thing to know is that, again, it provides compressive strength. So as an example, aggrecan is highly abundant within cartilage tissue, contributing to the ability to withstand compressive force in joints like your knee, like your shoulder, like your elbow, like your hips. To help envision what this looks like in tissue, we have a collagen fibril. We have a collagen fibril wrapping around here. That's in the darker gray. We have sorry, forgive me. Collagen, we have hyaluronin, and then hyaluronin is the dashed structure down the middle. And then we have aggrecan coming off the sides here, these gags. And so when there is compression, we see all of this being brought in close. We see all of this aggrecan being brought in close together, high negative charge, high repulsion, as well as water is being physically forced out by compression, creating a high osmotic pressure for water to go back in to this tissue. And those two things, the high negative charge and the high osmotic pressure, enable this tissue to resist compression and get back to its normal condition. Before we move further, I'll stop for a couple of questions here to answer any questions about fibroblasts or proteoglycans. Yeah. They produce collagen. Do they produce elastin? I was going to say, I'm trying to, in my mind, think about elastin-rich tissues where fibroblasts are typically expressed. So no, so I would not say fibroblasts. I would say typically smooth muscle cells are going to be producing, helping to contribute to elastin expression. Um, yeah. Um, but collagen is one of the um, is like one of the main proteins that fibroblasts express. Um, there's a school of thought that like different flavors of fibroblasts are meant to produce collagen, but that's like a whole active area of research. But yeah, collagen is definitely expressed by fibroblasts, as well as the different proteoglycans we talked about. Yes. Yes, they'll also produce laminin as well. Yeah. I'm going to try and do my best to make sure that I articulate what the take home is, because this is a lecture, at least for me, that it's very easy to go off the rails and talk about things that I find interesting that you guys might find terrifying uh, in terms of content and what to memorize. Yes. Mhm. Yep. That's one mode of resisting compression. I don't know if one mode is the right way to put it. One piece of the puzzle of how our tissue can resist compression. Yeah. 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 So if there is compression within certain cartilage tissue, that water is not being forced out of the tissue, but kind of like there's like displacement of the tissue itself sometimes under compression. So there's like a local osmotic pressure that's being driven here. Um, it's not necessarily totally leaving the tissue and going to other tissues, if that makes sense. And there's kind of just like this local displacement of water that creates an osmotic pressure to drive it back in. Yeah. All right, I'll keep moving forward. And again, if there's a question that percolates to your mind, please feel free to jump in mid-slide. So with a connective tissue ECM, we talked about proteoglycans. We're now going to talk about fibrillar proteins. The first fibrillar protein that we're going to focus on is collagen. So you can appreciate here that in the connective tissue ECM here, that with all of these, with these different cells here, that there is um, different flavors of proteoglycans throughout this connective tissue ECM, but as well, but what is also present throughout it are collagen fibers. Collagen is the most abundant protein in mammals. For those of you who have created just like bone broth at home, where if you take like a chicken or turkey carcass and just boil it down, you'll get like a gelatinous um, you'll get a gelatinous broth, for lack of a better phrase. That's because that gelatin is collagen. It's denatured collagen. And so 25% of all of the proteins in mammalian biology is collagen. It exists as three alpha chains wrapped together that form this triple helical structure that you can see in the images here. all collagen has a common motif, a common set of repeating amino acids. It's glycine and then X and Y. The reason why it's X and Y and not XX is because X and Y are usually different from each other. Usually that X is proline, and then usually that Y is hydroxyproline. Whenever you see hydroxy anywhere, and we're going to come back to this, that's just going to be an oxygen and a hydrogen together. So this is a hydroxyl group. So whenever you see hydroxy something, that's usually a hydroxyl group added to it. So in this case, Y is usually hydroxyproline. The reason why that's in blue and the reason why that's necessary is because collagen is the only protein in mammalian biology that has hydroxyproline. And what's distinct about that is we can actually use that for people who study collagen or want to study or measure collagen content in tissue. You can actually just do an assay to measure how much hydroxyproline is there. And so that's called the hydroxyproline assay. We do it regularly in labs that study scar formation and tissue repair. And so that's why it's noteworthy here. Hydroxyproline is only found in collagen. There are 42 different kinds or flavors of alpha chains. This slide is a little outdated. It says 28 collagens have been identified. I think we're at 35 or 33 now. And so I'm going to ask you to memorize every single one. That got a laugh. That got your attention. No. I could see the look of panic on your faces. Didn't mean to scare you. We're gonna talk about maybe two flavors of collagen, and that's really it. We've talked about one. We talked about collagen type 4 that's found in the basement membrane. I could tell that terrified all of you. Sorry about that. I was feeling a little spicy today. All right. So in addition to that, so that first amino acid glycine, if you remember from our amino acid lecture, we talked about glycine as having functionally no side chain. There's nothing at that alpha carbon at the amino acid for glycine. And so that makes it a hydrophobic amino acid. There's no charge on that side chain. And so what ends up happening is you can tell on the top right image here, we see X and Y labeled, but there's no glycine labeled because that's going to be buried within the backbone of collagen fiber. And so usually proline is going to be, both proline and hydroxyproline are going to provide stability. Again, because it is a kinking amino acid, it's going to produce that helical structure that it induces some stability. The event of hydroxylation typically happens in the ER. And so there's going to be, that's the endoplasmic reticulum. And so, excuse me. So prior to assembly of procollagen, we're going to have the addition of that hydroxyl group to make that proline, hydroxyproline. And there's a graphical image here of the structure on the bottom right here of collagen. And again, you can see glycine and white buried within. It's not always going to be proline and hydroxyproline. That would then make every single collagen species the same. You will typically find proline at the X position, and you will typically find hydroxyproline in the Y position, but it will not always be there. There might be other amino acids present. And so you can see in this image here that there is some arginine, there's some leucine, some threonine, but by and large, you will typically find proline at the X position and hydroxyproline at the Y. And again, hydroxyproline is only found in collagen. That hydroxylated proline is only found in collagen. Looking at different flavors of collagen, looking at the diversity of it, the most commonly found form of collagen is going to be collagen type 1. You'll find that in a variety of tissues, which is typically, which explains why it was the first collagen found, which is why it was dubbed collagen 1. The first three collagen types are going to be fibrillar. And those first three are going to be found across a lot of tissues that are present throughout the body. And in addition to its type, you can see how what its polymerized form looks like. If you move down, you can find collagen type 4. It's a sheet-like network. Again, that's a basement membrane associated species of collagen. And there are more recently discovered forms of collagen. There's a transmembrane form transmembrane form of collagen, as well as another form that's a core, serves as a core protein in proteoglycans. And so the key thing to note and walk away from this slide is there are lots of different types of collagen out there that are found in specific tissues. Some are abundantly expressed across a lot of tissues, for example, collagen 1. Some are a little bit more specific, with example of collagen type 28 here, or 18, forgive me. But again, the key thing to walk away here is that there are many different types of collagen, collagen 1 being the most abundant. And we can also characterize them or categorize them by their structure. So when we consider what the structural hierarchy is of collagen, they're not present in tissue as individual proteins, but rather there's a way in which they assemble together to provide real integrity to tissue. So if we think about the hierarchy, we first start with the alpha chain, which is, again, it's 3 alpha subunits together. So the alpha chain or alpha subunit is one single protein. When we think of pro-collagen, again, when you see pro, think of the precursor form. It has the pro cap. The pro-collagen it has a pro cap, and it's 3 alpha subunits together. Okay. Now, what's going to end up happening is when that pro cap is cleaved, we can now get, in some cases, we're going to get polymerization of collagen end to end in some forms of collagen. Collagen type 4, it's going to be more cross-hatching, more network-like. But with collagen type 1, the fibrillar kind, which is what we're going to focus on in this slide, fibrillar collagen, you're going to, once that pro cap is gone, you're going to get the self-assembly end to end. Now, when we get, now we'll get to collagen fibrils. It's important to distinguish fibrils versus fibers. So once we get to fibrils, they're going to start associating laterally. So at the pro-collagen step, there's a pro cap, and they're going to be assembling end to end. You can almost think of this, we talked about this a little bit last class. There's a cryptic site revealed that enables polymerization. But when we get to fibrils, we're now having this lateral association of collagen that's coming together. And when we get to the fiber level, that's taking fibrils that also, now imagine this is 10 times larger, fibrils that are associating laterally together as well, that makes up a collagen fiber. And so the image here helps us understand that, where we have specific alpha chains, that's one singular strand here. We have pro-collagen, which is 3 alpha chains woven together that are then going to end-to-end polymerize. We're going to have collagen fibrils, where these are laterally associating together. And then collagen fibers are fibrils that are also laterally associated together. And so this organization of collagen fibrils and fibrils is really important, because the orientation is going to be dictated by the kind of load your tissue experiences. So, for example, tendons typically undergo a uniaxial load. If you think of your bicep tendon, that's usually experiencing a load in just like one single direction. It's usually flexion of your forearm to your upper arm. So the collagen that's present in your tendons are typically all oriented in one single direction. Again, structure informs function here. Articular cartilage is going to be slightly different, as well as both bone and skin. Both bone and skin are typically going to be perpendicular layers of collagen together, and you can see that here in tadpole skin. And so the organization of collagen, because collagen's key function is to help provide tensile strength, its orientation will be determined or dictated by what kinds of loads or what direction of tension does that tissue experience. Another way in which these collagen fibrils can be stabilized to resist some of those tensile forces is actual cross-linking across collagen fibrils. And so, If we have two adjacent fibrils together, imagine I stapled these two markers together. That staple is then being cross-linked together. And so there's an enzyme that enables it, lysyl oxidase. Again, with enzymes, just break down the name and it's going to tell you exactly what it does. It's an enzyme that catalyzes a reaction, an oxidation reaction at lysines here. So again, we know that collagens have a glycine and then some X and some Y. The X's are a lot of times are oftentimes going to be some sort of, it's going to be proline, can be something different. And then Y's can be hydroxyproline, can be something different. In the cases where there's a lysine at X or Y, that's going to be a site at which lysyl oxidase, this enzyme, mediates a oxidation event that forms that changes lysine into an aldehyde by nature of that oxidation event. And now those aldehydes are incredibly reactive. We'll actually talk about that in particular when we get to metabolism and how their reactivity can actually be really bad in nutrition. But in the case of collagen here, that aldehyde can undergo a spontaneous reaction with other lysyl oxidases that produce aldehydes to then cross-link together. Okay, so this cross-linking mediated by lysyl oxidase changes lysine to aldehyde, and aldehydes will cross-link together here to form a cross-linking event that stabilizes collagen fibrils. So I'll stop here and give space for a few questions. Yes. Yeah, so in the hierarchy, I'm going to talk loudly so the recording picks my voice up. So I'm not yelling at you. Just want to make sure that's clear. So the tendon is oriented, this is all standing for collagen. You're only seeing collagen in these images. So with tendon, it's all oriented in one direction. And that's helpful because tendon typically experiences load or tension in one specific direction. Both skin and bone will experience load, not uniaxially. So to be able to withstand some of those forces, there's going to be perpendicular orientation of collagen that helps enable the tissue to withstand tension in those different directions. Particularly with bone and skin, what's a good way to say this? Particularly with skin, I mentioned really briefly this idea of Langer lines, how there's like actual like location specific orientation of how our skin can withstand tension. And so that orientation is going to be slightly different based on where you are, whether it's the forearm, the top of your forearm, or the back of your calf, or your upper back. That skin is going to be able to withstand forces differently because your body moves differently at those different sites. But by and large, they're going to be stacked perpendicularly. The collagen fibrils will be stacked perpendicularly on top of each other. Yes.
So going back to the presentation, like the demonstration we did last class with the forearm skin.
Yeah.
Is there like certain parts of the body, like the forearm, where there's more collagen stacked in one direction than there is in the other? Because like how there's certain elasticity going one way and the other way has less elasticity.
Yeah, I mean, kind of similarly with the first thing that comes to mind is like with your thigh. Like you can pinch the skin in one specific orientation because typically that skin undergoes stretching in one direction when you squat down, but your skin typically won't experience a ton of stretch in the other direction. So there's going to be more orientation of collagen in your the dermis of your thigh, oriented more this direction, but there's still going to be collagen associated this way to help provide still some stability and tensile strength there. Does that help clarify? Yeah. Yes. The cross-linking is strictly a way to stabilize collagen fibrils together. So when and this is typically a feature of kind of mature collagen that's been there for a while. If you wanted to look at cells that are, I'm starting to get into my research, forgive me. The key thing to take away about the stabilization by cross-linking is that it is another method to further provide stability and strength to collagen fibrils to help them carry out their function, to help provide the ability to withstand tensile forces in a variety of tissues. The orientation of that cross-linking, again, will be different based on what tissue you're in. But if we zoom out and consider just collagen in tissue in general, that cross-linking is really important to just stabilize and provide more tensile strength. Yes, one collagen fiber is many, I'll go take a step back. One fiber right here, is many fibrils together. These collagen fibrils will be cross-linked together to further stabilize all of these fibrils together, as opposed to them being able to slide individually. With mature collagen fibrils, there's going to be lysyl oxidase at lysines that mediate an oxidation event that turns them into aldehydes, and these adjacent aldehydes will just react and cross-link across each other to stabilize these fibrils within a given collagen fiber. Yeah. All right, we will press on, but again, if a question percolates, go ahead and feel free to jump in. Okay. In addition to collagen, we also have elastic fibers that are comprised of a key protein called elastin. Elastin, it's an incredibly hydrophobic protein, and that's going to help mediate its function here. Similar to collagen, it has a common repeat here. There is no glycosylation that happens with elastin. There's not going to be hydroxylysine here, but there will be some hydroxyprolines present within elastin. So it's going to be far, far fewer than what collagen typically has. And so it still would be largely accurate to say that the chief protein that possesses hydroxyprolines is collagen, but it can occasionally be found within elastin as well. While most proteins in their precursor form are going to be have a prefix with as pro, elastin's precursor form is called tropoelastin. What's important to note here is that elastin, mature elastin, is typically very hydrophobic. That's not the case with its precursor form. So there's something about tropoelastin's pro cap that imbues it with a soluble, enables it to be soluble, and then it can be secreted into extracellular space and then be cross-linked similar to collagen. But it's when it's mature that tropoelastin is converted into elastin that it becomes hydrophobic that mediates its function. That function is that the hydrophobic nature of elastin contributes to its elastic properties. When our skin, I'm gonna keep going back to skin. When you apply tension to the skin, the ability for your skin to go back to its original shape is mediated by elastin. If there was a defect in elastin, when I pinch my skin, it would stay like that and just kind of like fall to the side, and now my skin would kind of look like this, for lack of a better phrase. It's also fair to say that, unfortunately, as we age, we lose elastin content. And so in older populations where you notice their skin sags a little bit more, that's because there's a loss of elastin content there in their skin. That becomes far more deleterious in other tissues, like specific blood vessels, where a loss of elastin can have really fatal repercussions. But that's just one visual description as to what it looks like when we start to lose elastin content in tissue. There's going to be alanine and lysine-rich alpha helical segments within elastin, and that's going to be where the cross-linking can happen for elastin. When we think about what tissues are rich in elastin, we want to think about what tissues where it's incredibly important for their function that when they undergo tension or undergo some sort of displacement, that they go back to their original shape. That is incredibly important for arteries, in particular your aorta. Your aorta is the segment of blood vessel that's just outside your heart, just outside the left ventricle, that experiences the greatest amplitude of pressure when you have a heartbeat. And so its ability to displace and return back to shape is incredibly important. And so you'll find that at the aortic arch, just outside your left ventricle, there's a high elastin content there. And you can see electron micrographs of a cross-section of an artery where when we zoom in below with this electron micrograph, we can see elastic fibers there. The way they are oriented, these elastic fibers, is that there is a core of elastin in the top, depicted in the top right here. And there's going to be fibrillin, that is a glycoprotein that's present all over the surface of elastin. So again, elastin is highly hydrophobic, so it's buried on the inside and camouflaged, for lack of a better phrase, by fibrillin. But it's labeled microfibrils there, by fibrillin proteins there. And what ends up happening is that in a relaxed state, we have elastin. I'm going to talk loud for the recording, so again, I'm not yelling at you guys. We have elastin in a relaxed state here, where it's buried on the inside. When it undergoes displacement, where there's stretching that happens, it exposes these elastin molecules. And what ends up happening is that because of its hydrophobic nature, it's going to want to recoil and go back to its original relaxed shape, the conformation where it's buried. And so it's energetically favorable for it to go from the stretched condition to the relaxed condition. Okay. And so this nature of elastin to provide elasticity to tissue is mediated or enabled by its hydrophobic nature. The final fibrillar protein we're going to talk about is fibronectin. What's unique about fibronectin is that it does not quite possess the same structural function that elastin and collagen have. This ECM protein is much more involved with, it serves a role as a bit of a cell signaling hub, because what you could appreciate here is that there are domains that bind to heparin, as well as some other gags, but heparin is chief among them. And if we remember from earlier, heparin is a growth factor depot gag. So what ends up happening is that heparin will bind there in fibronectin, growth factors will now bind to heparin, and so the cells that see fibronectin will also see growth factors that trigger cell signaling, trigger their activation. And there's really a wide-ranging series of events that can happen where growth factors binding to fibronectin or binding to heparin on fibronectin can mediate cell adhesion, cell migration. Again, cell adhesion is just the binding of the cell there. Migration is movement of cells. Proliferation is the growth of cells, the doubling events of cells. And differentiation is this specialization of cells towards a specific flavor of cell with a specific function. Fibronectin can be a ligand for, we talked about integrins last class. Integrins are the receptors on cells for ECM proteins. So fibronectin can bind to integrins on cells, in particular fibroblasts, again, because they secrete fibronectin and they also exist in the connective tissue ECM where fibronectin is found. And so fibronectin can really dictate a lot of functions of fibroblasts. We have a lot of domains present on fibronectin. What's interesting is that there's a fibrin binding domain. So fibronectin can kind of help serve as the, when you have an injury, you get a fibrin clot that forms. Fibronectin is laid down to help serve as this like early extracellular matrix during wound healing before collagen comes in, and it lays the tracks for collagen to be deposited by fibroblasts. We have collagen binding domains, and then chiefly, I'll stay here. This RGD domain right here is a classic amino acid residue that cells recognize via their integrins. And so the RGD domain is like the key cell binding domain based on that amino acid residue. Yes. It can be a what? Yeah, so a ligand, ligands bind to receptors. So it binds, cells can bind to fibronectin by their integrins. In addition to that, and one way to think about this is that if we, actually, I don't need to go back. We have a heparin binding domain at the very top. So when a cell binds to when a cell binds to fibronectin by its integrin, there's also going to be adjacent or nearby or close by some growth factors of that heparin binding domain, if heparin is there. And so there's going to be some degree of cells seeing growth factors that trigger cell signaling that will elicit some either adhesion, migration, proliferation, or differentiation. Does fibronectin migrate or? No, so what'll end up happening is that fibroblasts will secrete fibronectin. They'll express it. For example, during, I'll keep going to like during the wound. You have a blood clot that'll form. Fibroblasts, via their integrins, will recognize fibrin. And there are specific integrins that will recognize fibrin that trigger, once they see fibrin, they know the next step in wound healing is to secrete fibronectin here. And so they'll secrete fibronectin there on that fibrin clot. And so fibronectin now is able to trigger and bind to certain growth factors that, again, all of these things are actually really important for wound healing. So there's going to be the triggering of cells migrating because they bind to certain growth factors that are bound to fibronectin. There's going to be differentiation, proliferation. There's a when I say it's a cell signaling hub, what I mean by that is that there are cell signaling factors, growth factors, that can be anchored onto fibronectin because those growth factors bind to heparin, and heparin can bind to fibronectin. Trying to think if there's like a helpful analogy to illustrate a little bit more. Well, I'll just leave it there. Does that help clarify? Okay. So a key thing, so there's a lot of information here about fibronectin. To reiterate a few key things. It doesn't provide much structural integrity to tissue. What it does provide is lots of opportunities to trigger cell signaling. And so the ability for growth factors to bind to heparin is really important because that can mediate some cell signaling. The ability of cells to bind to these integrin binding domains at, there's different types of, there's different units or modules here. The oval ones are called type 3. So this 9th and 10th type 3 is really important for integrins. Those are the main integrin binding domains. And as you can see, other proteins or other ECM proteins like collagen, like fibrin, can bind to fibronectin. So its function is not providing structural integrity, but triggering downstream biological or cellular events based on its ability to bind to other molecules. So I've talked about integrins a couple times. They are the receptor on cells that bind ECM proteins. I'll actually push pause here and give space for any questions about fibronectin or anything we've talked about up to this point. Yes. I love backtracking. That's okay.
But I thought you said that collagen was in the protein with proteins, but then a later slide said that elastin also.
Yeah, so by and large, it's chiefly collagen. There is a very small amount of hydroxyproline present in elastin, but predominantly, if you were to measure the content in some like tissue sample, or if you were to separate out elastin and collagen and looked at how much hydroxyproline content was there, it is far more abundant and a more accurate way to say would be it is predominantly abundant in collagen, present in small amounts in elastin. I appreciate you asking that. I appreciate you backtracking. All right, so we've got 10 minutes. I think I can wrap this up. So integrins are the main receptors on cells that bind to ECM proteins. Integrins usually have an alpha subunit and a beta subunit. Those alpha and beta subunits come in different flavors, and they can be, there's different combinations of alpha units and beta subunits. So in humans, we have about 24 different combinations found on cells. And what ends up happening is, this is going to help kind of combine a couple of different concepts we talked about in the class, is that when our integrins bind to an ECM protein, that's going to produce a conformational change that allows a protein on the intracellular part to bind to the beta subunit. It's called talin. From there, talin is connected to our actin cytoskeleton. So integrins are a way of connecting the outside of the cell to the inside of the cell. That ability to connect and bridge the extracellular matrix to the actin cytoskeleton of a cell helps provide mechanical support for the cell, and is actually a really fascinating way to induce signaling via some mechanical force. So again, this is mechanobiology. I've referenced it a couple of times. I'll probably likely come back to it a couple more times. But the ability to anchor but this is, again, combining a couple of concepts here. Integrins bind to the ECM. It induces a conformational change in integrin. And what that looks like, I think it's on the next slide. No, it'll be 2 slides from now. There's a conformational change induced, and that will actually enable the ability of talin to bind to the beta subunit and bridge and connect to the actin cytoskeleton here. There is this conformational switching of the integrins. So when they are inactive, they're kind of, imagine someone has taken if we take a step back, a crude way to describe integrins are that each subunit looks like almost like a golf club. And the head of it is where it binds to the ECM. In its inactive conformation, imagine someone has taken that golf club and just bent it at, say, 45 degrees right in the middle. So it is typically inactive in a bent inactive conformation. Again, imagine a golf club bent in half. But there's allosteric regulation where if the ECM binds to that bent golf club head, it will now open up, and those 2 subunits will separate a little bit, and now there can be space for intracellular signaling. The integrins traverse across the entire cell membrane, where we have these ECM binding domains outside and these intracellular domains on the inside of the cell that can help mediate this outside-in signaling. And that's actually one of the next things that we'll talk about, is this outside-in versus inside-out signaling of integrins. But again, the key thing to walk away with here is that there's active and inactive conformations of integrins. Again, just visually think of like a bent golf club that can unbend and be totally active and straight if it binds to the ECM. But there's also ways in which that can change based on inside-out signaling. So first, outside in, we'll have integrin in its bent inactive conformation. There's actually rotary shadowing images here of the inactive and active. Again, that RGD, that integrin binding part of fibronectin, you can just synthesize that amino acid and that peptide and put it in cell culture and see how it affects cells. So it's inactive. When it's in the presence of some ECM protein or some integrin binding domain, what ends up happening is that induces a conformational change. When the head of integrin binds to the ECM, there's a conformational change. Now, this beta subunit of integrin is able to bind to talin and trigger downstream events by nature of also being bound to the actin cytoskeleton. That's outside in, binding to the ECM, conformational change of integrin, binding to what's on the inside of the cell, in this case, talin and then actin. There are instances where there's inside out. Talin is competing for this beta subunit. The alpha subunit's in the way, but it's trying to compete and get access to it. There are instances where there are events where talin can actually bind to the beta subunit. And once that happens, those subunits will now come apart. Again, this is originating from inside. There's on the previous slides, there's talin depicted. And so it's competing for the beta subunit. Inside out, the key thing to note is talin, the presumption for inside out is talin is always competing for the beta subunit. It wants to get there. Again, because this is like my life at home, like imagine a kid just like, I want to go sledding, I want to go sledding, I want to go sledding. There's no snow outside. And then finally, there's like some weird event where snow happens. It's a broken analogy, forgive me. But now Talon can actually bind to the beta subunit. And now that's going to trigger a conformational change that opens up the integrin to now be able to bind to the ECM. So there's outside in, it starts on the outside because the ECM binds. That's the presumption for outside in. The presumption for inside out is Talon is competing for this beta subunit. And it wants to bind. It's that kid that wants to go outside and go sledding. Once it finally does, it triggers conformational change, and now integrin can bind to the ECM. Are there, I'll give space for a couple of questions here. Yes. What's the purpose of the integrin binding to the ECM? Like all of biology, man. So, the ability of a cell to bind to its ECM is critical to a fetus developing into a fully formed human. The ability of organ development, because the ECM is basically the environment in which all cells live, and they take cues from the ECM. So the ability to engage with the ECM drives, I talked last class about stiffness, being able to induce embryonic stem cells to become some specific cell. The stiffness of the ECM provides that cue. That's like a biophysical cue. The biochemical cue of the ECM is what proteins are there, what growth factors are anchored to the ECM, and what is the orientation of the ECM that can dictate not just if it moves, but how it moves. Does that make sense? I mean, honestly, again, like in a 2-day-old blastocyst, we're already getting ECM expression. We're already getting laminin expression. So there's during development, during morphogenesis. Morphogenesis just means like the development of different tissues and organs. All of it is, I'm a little biased. All of it is mediated by the ability of cells to use their integrins to bind to the ECM. Again, if you knocked out in the first half of this lecture, if you knocked out specific ECM proteins, it's embryonically lethal. If you knock out specific integrins, there's research that shows it's embryonically lethal. So the ability of an integrin to bind to the ECM, your life depends on, both during development and now. Sorry to be melodramatic. We have like 2 minutes left. I'm going to press on. So we've been talking about the production of the ECM. We've been talking about how it's organized, how it's stabilized. But so far, we have all production of ECM proteins. It's all gas. We need the brakes. We cannot just be creating or synthesizing ECM protein all the time, or else we would all just be one big mass of collagen. Or more, in a more clinically focused way, our organs would just be full of scars. We would all be, our organs would be full of fibrosis. And so there needs to be breaks, ways to degrade ECM, to regulate it and make sure it's not just constantly being synthesized. So we call the degradation of the ECM proteolysis. And there are 2 main flavors of enzymes that degrade the ECM. We have matrix metalloproteases, or MMPs, and we also have serine proteases. Matrix metalloproteases, they're called metalloproteases because they rely on some specific cation that serves as a cofactor for them. Cofactors are some molecule that helps enable the function of an enzyme. So there's going to be, if you look at the list of metalloproteases, or MMPs here, you'll be able to look them up and see like what specific metal ion they are reliant upon as a cofactor. And then to the right, you'll see what are the substrates that they act on to degrade that ECM protein. So there's an image here that shows degradation of a ECM protein. This is enabled during tumor development, during morphogenesis. All of this is key. It's a balance of gas versus brakes, production of the ECM, and then degradation of it. It's always a constant balance that our body's trying to strike so that our body has sufficient strength and that also that we're not undergoing constant scar formation or constant collagen production in different tissues and organs. There are different mechanisms that regulate this, and I'm going to make this the last slide. Yeah. I'm the second to last slide. We have local activation. That's we have specific precursors that activate these MMPs. There can be receptors on the surface that regulate them. And when we talked about the pro-cat being cleaved on collagen type 4 and laminin, that's MMPs on the surface regulating these proteins. And there's inhibitors of these metalloproteases called TIMPs, tissue inhibitors of metalloproteases. So that's the brakes on the brakes. When tissue engineering or regenerative medicine uses the ECM, we can take tissue, remove all of the cells, and you just have the ECM left. And that's the basis for a lot of biomaterial scaffolds or tissue engineering scaffolds to create new tissue, where it's the blueprint that cells engage with by their integrins to promote tissue regeneration and repair. Or there are biomaterials or implants that are meant to mimic the ECM, providing that scaffolding, that those biophysical and biochemical cues to promote tissue repair and tissue regeneration. I'm going to skip over a ton of slides and just go to the end. Nope. I lost it. There's some folks I was going to recommend. If any of this fascinated you, I highly recommend you check out the folks at UVA that are in this space. Don Griffin, Stephen Cagliari, Chris Hiley, Kali Lampe, Rachel Lettieri. There's also a lot of folks across the country doing this work. Thanks for your time. I'm 2 minutes over. Sorry about that. Have a good one. And I will see you for our exam and quiz on Thursday. Quiz first. If you are happy with your quiz scores, you'll then be dismissed and we'll move to the exam. Thank you.
Thank you.
Yep, you have a good one.